So I wanted to do a video on sirtuin activation. Sirtuins are a very popular target for slowing the aging process, and we're going to see why. So essentially, I want to first cover what sirtuins are. So sirtuins are signaling proteins involved in metabolic regulation. Okay, I'm going to skip through this pretty quickly because I'm a little pressed for time today. Actions. Sirtuins are a family of signaling proteins involved in metabolic regulation. They are ancient in animal evolution and appear to possess a highly conserved structure throughout all kingdoms of life, whereas bacteria and archaea encode either one or two sirtuins. Eukaryotes encode several sirtuins in their genomes. In yeast, roundworms, and fruit flies, Sirt2 is the name of one of the sirtuin-type proteins. Mammals possess 7 sirtuins, Sirt1 through Sirt7. Okay, so that occupy different subcellular compartments. Sirt1, Sirt6, and Sirt7 are predominantly in the nucleus. Sirt2 is in the cytoplasm, Sirt3, Sirt4, and Sirt5 in the mitochondria. Sirtuins and fibrosis. A 2018 review indicated Sirt levels are lower in tissues from people with scleroderma, and such reduced Sirt levels may increase risk of fibrosis through modulation of the TGF-beta signaling pathway. DNA repair. Sirt1, Sirt6, and Sirt7 proteins are employed in DNA repair. Sirt1 protein promotes homologous recombination. Now, this is why DNA repair is, of course, very important to longevity and overall health. So we're going to talk about this in a little bit later. Sirt6 is a chromatin-associated protein, and in mammalian cells is required for base excision repair of DNA damage. Sirt6 deficiency in mice leads to degenerative aging-like phenotype. In addition, Sirt6 promotes the repair of DNA double-strand breaks. And I'll go over double-strand breaks when I cover Maria Konovalenko, because she did a beautiful video on double-strand breaks. Okay, stimulate homologous recombination repair. Sirt7 knockout mice display features of premature aging. Sirt7 protein is required for repair of double-strand breaks by non-homologous end joining. Inhibitors. These are some of the inhibitors. Okay, sirtuin activity is inhibited by nicotinamide, but we'll explain why that's important, which binds to a specific receptor site. It is an inhibitor of in vitro of Sirt1, but can be a stimulator. That's funny, they misspelled stimulator in sauce. Activators. Pinsa, whatever, that's like, I believe that's marine pine bark that stimulates Sirt1. This Sirt1, cyanide, Sirt6, obviously don't be taking cyanide. Proanthocyanins, that's from berries. Quercetin and rutin derivatives from citrus and citrus peels. Luteolin, Sirt6. Catechin and epicatechins from cocoa and green tea, Sirt6. Fisetin, Sirt6. Phenolic acids, Sirt6. Whatever the hell, Sirt6. Curcumin, Sirt1 and Sirt6. All these different chemicals, these stimulate Sirt1, Sirt6, etc. So I'm going to move on. Sirtuins, a promising target in slowing down the aging process. These are some of the compounds that stimulate sirtuins. Of course, we've already mentioned some of these. Of course, resveratrol, quercetin, butene, fisetin, camphorol, whatever. Catechins, which are again from cocoa and green tea. Proanthocyanins, which are from cocoa, berries, things of that nature, purple sweet potatoes, etc. And as I said, of course, resveratrol. Supplementation of polyphenols may protect against neurodegenerative, cardiovascular, inflammatory, metabolic diseases, and cancer by enhancing Sirt1 deacetylase activity. However, in humans, the therapeutic and pharmacological potential of these natural compounds remains to be validated in clinical conditions. Now, I want to make sure people understand I do plan on doing a very heavy-duty video on this, but right now I'm going to somewhat rush through this just so I can get done with it, but I do plan on doing a much more intensive video on sirtuins. not related to it'll mention phytonutrients, but other things also. Physical activity. Regular physical training is able to improve the quality of life. Exercise improves the resistance of oxidative stress. This is again through hormesis. What this does is the stress caused by exercise, the oxidative stress, actually enhances sirtuin activation and DNA repair and things of that nature. I generally would like people to follow this article. This is a very well put together article, and I strongly encourage people to read it. This is about curcumin and sirtuin regulation, and I'm going to get into this much deeper in a later video, but for now it will suffice that I'm just covering some of the basics here. Sirtuin signaling in cellular senescence and aging. Anti-aging effects of sirtuin. Cellular senescence is a physiological phenotype aimed at permanent cell cycle arrest and is morphologically identified as flattening, increased size of nucleus and nuclei, and the appearance of vacuoles in the cytoplasm. In addition, several biomarkers developed for cellular senescence are targeted towards the senescence associated with beta-galactosidase, telomere attrition, senescence-associated blah blah blah, hemochromatoin, whatever. Although still under debate and not fully defined, growing evidence have shown that sirtuin is an essential factor in delaying cellular senescence and extending organismal lifespan, meaning it extends the lifespan of various organisms. Remember, these are very ancient aspects of life. Okay, they go back to bacteria all the way through the animal kingdom. Okay, the role of sirtuin on the protection for cellular senescence has mainly been investigated with mammalian SIRT1 and SIRT6. The levels of sirtuins, including SIRT1 and SIRT6, but not SIRT2, are reported to decrease in senescent cells of mouse embryonic fibroblasts, lung epithelial cells, human endothelial cells, and macrophages exposed to oxidants. In addition, the reduction of SIRT1 and SIRT6 using pharmacological inhibitors, siRNA and miRNA, promotes premature senescence, like phenotype, meaning it causes whenever you see premature senescence or senescence, think of senescence as aging. That's what it means essentially. Conversely, the overexpression of SIRT1 and SIRT6 suppresses the cellular senescence, so it suppresses aging. The sirtuin-related suppression of cellular senescence is mainly mediated through the prevention of telomere attrition and the promotion of DNA repair. Sirtuins play vital roles in sustaining genome integrity, meaning they keep your DNA structure healthy by contributing in maintaining the normal chromatin condensation state and responding to DNA damage and repair, especially the nuclear form of sirtuins such as SIRT1, SIRT6, and SIRT7 act as transcriptional regulators to suppress gene expression by stabilizing the chromatin structure. SIRT1 deacetylates histones H3 and H4 and H1, and more than 50 non-histone proteins. Okay, SIRT1, SIRT6 are known to regulate the expression of telomere reverse transcription required for telomere elongation and to deacetylate histone 3 lysine. Okay, sirtuin and organismal lifespan. In addition to the roles in cellular senescence, it is well established that sirtuin regulates organismal, meaning the lifespan of organismal lifespan in several animal models. Increased expression of levels of sirtuin, especially yeast SIRT2 and its homologues, extends lifespan of budding yeast, worms, fruit flies, mice. The first investigation of the pro-longevity effect of SIRT2 was established using the yeast model system almost 20 years ago, in which the complex blah blah blah. Okay, let's see. Sirtuins other than SIRT1 are also reported to exert pro-longevity effect. The transgenic male mice overexpressing SIRT6 showed a significantly longer lifespan than wild-type mice, by 16%, whereas SIRT6 and SIRT7 deficient mice live shorter than controls. In addition, the polymorphism in SIRT3 has been reported in European centenarians. In Drosophila, which is fruit flies, the overexpression of DSIRT4, which has a mitochondria targeting sequence in the whole body or fat body, was reported to extend the lifespan and increase the resistance to starvation. The expression of DSIRT4 was induced by starvation in the fat body. This is also, you have to understand, caloric restriction also stimulates sirtuins. The molecular targets of this longevity effect of sirtuins have been actively investigated. Sirtuins are found to especially interact with all the major conserved longevity pathways, such as AMP-activated protein kinase, which is AMPK, insulin, IGF-1 signaling target of rapamycin. So these sirtuins affect all of these other genetic and metabolic pathways, FOXO in worms, blah, blah, blah. Lifespan extension by overexpression of constitutively active DFOXO in adult fat body was considering FOXO is a major component. Let's see here. In addition, deletion of okay, let's see. Let me AMPK signaling belongs to protein kinase family and restores cellular energy levels. Increased AMPK activity is known to extend lifespan of some model organisms. The mutation of AMPK in nematode worms abrogated the lifespan extension by SIRT2 expression, indicating that AMPK also contributes to sirtuin-induced lifespan extension. SIRT1 activates AMPK through the direct deacetylation of LKB1, a regulator of AMPK, and AMPK is known to activate SIRT1 through the elevation of NAD+ levels. In addition, AMPK contributes to the pro-longevity effect of the IIS, suggesting that these longevity pathways intricately crosstalk with one another. Caloric restriction. Now, I want to make sure that people understand caloric restriction with optimal nutrition, such as was supported by Dr. Roy Walford, is the ideal, not simply just cutting calories. You need to maximize the micronutrient. You have to maximize your micronutrient, optimize your micronutrient intake, and also make sure you're not deficient in fatty acids or amino acids or any of the other things that you require. But remember that animal models, including non-human primates, which experimentally means a reduction in caloric intake by 10 to 50% compared to ad lib intake without malnutrition. Although still controversial, it is believed that the beneficial effects of caloric restriction on lifespan extension and prevention of age-related diseases is mediated by the induction of sirtuins. In many animal models, the expression and activity of sirtuin was reported to be increased by calorie restriction and nutritional deprivation. That's the problem, is a lot of people refer to caloric restriction as undernutrition. It is not undernutrition in the sense that you're reducing calories, but you're not reducing calories so far that you're inducing starvation. You're reducing calories enough to activate these pathways, but you're also taking in enough of the micronutrients, especially in the practice as promoted by Roy Walford. You do not want to affect issues with the, like for example, the triage theory, which I'll get into with Bruce Ames. You don't want to cause issues with, deficiencies in certain vitamins, certain minerals, certain amino acids, certain fatty acids. You need all of these things. So sirtuin by caloric restriction was mediated by the upregulation of AMPK and increase of NAD+ levels in nematode worms. The expression levels of SIRT2 tagged with M-cherry fluorescence increased due to starvation in the intestine. So these animals, of course, these worms are being starved, but obviously if you're doing this, you're not going to starve yourself. You're going to practice caloric restriction with optimal nutrition. It also, expression increased in flies fed low-calorie food. In mammals, the levels of sirtuins were also reported to increase after caloric restriction, which is known to be tissue-specific. Increased levels of SIRT1 by caloric restriction were observed in white adipose tissue, skeletal muscle, kidney, brain, and intestine. However, in liver, the response of SIRT1 expression to caloric restriction is debatable. One paper showed induction of SIRT1 in the liver of caloric restriction experienced mice, but another report showed a reduction in levels. Furthermore, the latter report also showed that knockout of SIRT1, meaning animals that have that taken out of their genetic code in their livers, is dispensable in this tissue. In addition, SIRT6 is indirectly activated by caloric restriction by upregulating SIRT1, the FOXO3, alpha, the nuclear respiratory factor, NERF, I believe that's NERF1. SIRT2 and 3 are induced by caloric restriction in diverse tissues, including muscle, white adipose tissue, liver. SIRT3 is suggested to be an essential for cochlea neurons against oxidative damage. These studies indicate that the expression of sirtuin is regulated by caloric restriction with tissue specificity, suggesting that a more elaborate investigation is required to understand sirtuin regulation by caloric restriction. Activators of sirtuin. Since sirtuin is commonly believed to mediate the beneficial effects of caloric restriction, the activators of sirtuin are considered to mimic these beneficial effects and are hence attractive therapeutics for age-related diseases. Remember that it was determined by the Wilcox brothers that the Okinawan diet, while it has mild caloric restriction, it also seems that a lot of what are called caloric restriction mimetics, or sirtuin activators would be another way of saying that, but caloric restriction mimetics are most likely extremely responsible for the health benefits of the Okinawan diet. So some of these molecules include quercetin, butene, fisetin, pisatinol. Like I said, that's I believe that's marine pine bark. The most potent activator of SIRT1 to be resveratrol, a polyphenol found in red wine, which extended the replicative lifespan of budding yeast by 70%. Addition, the lifespan extension effect of resveratrol was abrogated by the SIRT2 mutation, indicating that resveratrol extends the lifespan of the yeast through the activation of sirtuin. Interestingly, higher concentrations of resveratrol did not further increase the lifespan, and resveratrol failed to extend the chronological lifespan. Another study in the following year showed that fisetin, butene, and resveratrol also activated levels of sirtuin-2 of nematode worms. In addition, single amino acid of SIRT1 was proved to be critical for binding STACs and inducing activation. Supplementation of resveratrol at 100 nanomoles, I believe that is, extended the lifespan of nematodes and I don't know what that animal is, by 14 to 29% respectively, but was ineffective on SIRT2 mutant, suggesting that resveratrol extends the lifespan in SIRT2 dependent manner. However, the lifespan of transgenic worms overexpressing SIRT2 were extended by 39% following resveratrol treatment, thereby suggesting that SIRT2 exerts its effect of extending the lifespan independent of resveratrol. In addition, the study also revealed that resveratrol-induced lifespan extension is mediated by the ER stress gene. Expression, regulation, other than resveratrol, natural compounds including silozazole, painol, statins, hydrogen sulfide, icrian, persimmon, melatonin, and curcumin are also reported as potent STACs. Some of these STACs have exhibited a pro-longevity effect on model animals. For example, pre-treatment with curcumin or whatever, enhanced SIRT1 activity and extended lifespan of fruit flies. Natural STACs are hydrophobic in nature with low solubility and low bioavailability. To overcome these weaknesses, synthetic STACs were developed by using drug design approaches. To date, more than 14,000 STACs. Another way to make them more soluble is to take these substances with lecithin Lecithin is an emulsifier. These synthetic STACs are reported to be beneficial for several age-related diseases and have demonstrated protection from cancer, neurodegeneration, cardiovascular disease, and diabetes, with some compounds exerting an extended lifespan of these. These are the different synthetic drugs: sirtuin 1720, sirtuin 2104. SRT 1460, SRT 2183, STAC 5, STAC 9, and STAC 10. These are the synthetic compounds that were created to stimulate these pathways. Okay. An alternative approach to activating sirtuins is regulating NAD+ levels by activating enzymes involved in biosynthesis of NAD. NAD is an essential cofactor for, this is a form of niacin, is an essential cofactor for electron transfer and for regulating metabolic homeostasis, and its levels decrease with age in the liver and muscle, as could potentially be explained due to the decreased activity of sirtuin upon aging. The supplementation of NAD, extended lifespan of both nematode worms and fruit flies. During deacetylation reaction by sirtuin, NAD+ is converted to nicotinamide, and nicotinamide is recycled into NAD or other nicotinic acid derivatives, blah, blah, blah. For over 20 years, the sirtuin family has been actively investigated for its function in delaying cellular senescence and extending longevity. In addition, based on the role of sirtuin on the beneficial effect of caloric restriction, therapeutic trials using activators of sirtuin have actively proceeded to protect age-related diseases. Growing evidences have principally supported that sirtuin is an attractive anti-aging molecule involved in improving health through the target molecules participating in diverse biological processes. However, the role of sirtuin on longevity and the longevity effect of caloric restriction are still controversial. In addition, numerous questions remain unresolved, such as the role of other sirtuins, in addition to SIRT1 and SIRT6, on aging and redundancy of sirtuin family members to regulate lifespan, whether other enzymatic activities apart from deacetylation activity participate in process of aging, and whether STACs could be promoted as drugs to treat aging or age-related diseases in humans. These questions will be answered in the near future, and sirtuin may provide the effective approach to extend lifespan and improve quality of life. So this is about about activating sirtuin 1 with natural phytochemicals. And this is what you have to understand. This is in my video on how to extend human lifespan for real. I talk about the role of phytonutrients in extending human lifespan and why eating plants is healthy. The reason why is because it modulates numerous activities. Substances, these phytonutrients, modulate DNA repair, metabolism, oxidative stress damage, blah, blah, blah. But what you'll see is polyphenolic compounds, polyphenolic products in fruits, vegetables, and plants, including resveratrol, quercetin, and curcumin, will be discussed in this article. But the thing that you have to understand, these substances that are in fruits, vegetables, etc., particularly spices, citrus peel, things of that nature, these help to encourage DNA repair, healthy metabolism, and reduce oxidative damage, things of that nature. Natural polyphenols as sirtuin 6 modulators. This is from Nature magazine. Flavonoids are polyphenolic secondary metabolites synthesized by plants and fungus with various pharmacological effects due to their plethora of biological activities. They have been studied extensively in drug development. They have been shown to modulate the activity of NAD+ dependent histones, SIRT6, because SIRT6 has been implicated in longevity, metabolism, DNA repair, and inflammatory response reduction. It is an interesting target in inflammatory and metabolic diseases as well as in cancer. Here we show that flavonoids can alter sirtuin 6 activity in a structure-dependent manner. Catechin, which again, cocoa and tea derivatives, potency against SIRT6, blah, blah, blah. Introduction. Flavonoids are a large family of naturally occurring phenolic polyphenolic compounds that provide important health benefits and help to protect against cancer, cognitive decline, diabetes, heart disease, and obesity. Chemically, flavonoids contain a 15-carbon skeleton, blah, blah, blah. Not important for our purposes here. Nobody wants to know about that. The antioxidant property of flavonoids may be mediated by many mechanisms, including inhibition of enzymes involved in free radical generation and subsequently suppression of reactive oxygen species, what we commonly call free radicals. Sirtuins is an enzyme family that can modulate, or basically free radicals. They help with DNA repair. Resveratrol is widely studied. The thing is with resveratrol is widely studied, but do not think that resveratrol is the only thing that does this. I take a ton of quercetin, and quercetin seems to work well by me, better in fact than taking resveratrol did for years. I'm just putting that out there. I don't know what the research is on that, but I know quercetin really has seemed to benefit me quite well compared to resveratrol. Flavonoids modulate SIRT6 deacetylation activity in vitro. A set of flavonoids and phenolic compounds were tested, blah, 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 2 substrates, yada, SIRT activity was determined. Let's see what we got going on here. The observed, let's see. Catechins, again, this is from green tea and cocoa. Catechins and epicatechins are stereoisomers that results in different inhibition towards SIRT6. Catechins with transconfiguration are more potent SIRT6 activators than the cis version. Flavonones, okay, did not improve the inhibitory activities towards SIRT6 compared to the flavonoids and flavanols. Compound was a weak inhibitor and it increased deacetylation. Anthracyanids, these are in berries, pomegranate, and again cocoa. So what did they do? They both showed an increase in deacetylation activity for SIRT6 compound with significantly more effective, producing 55-fold maximal activation compared to other activators. So that's berries, cocoa, purple sweet potatoes, those sorts of things. Isoflavonones, these are from seeds, beans, other legumes, beans, like lentils, soy, etc. Was able to activate SIRT6. Phenylitic acids, a set of phenylitic acids, gallic acid derivatives. That's in pomegranate peel, etc., which is another main class of plant polyphenols, were also included in the study, although compounds 22 and 23 increased slightly SIRT6 activation. Overall, phenylitic acids were weaker modulators than flavonoids. Remember that. Instead of buying high sugar pomegranate juice, it might be better just to have a couple cups of cocoa a day, or maybe a blueberry smoothie that has less sugar and has the procyanids, or let me see if I can pronounce that again, cyanidins, which are in blueberries, that probably is going to have a better effect. Okay. Among SIRT2 and SIRT6 has been implicated in aging and age-related diseases because its physiological role is not completely understood. The extent to which increased SIRT6 activity affects these disease conditions is still unclear. It might offer a protective mechanism or alternatively represent part of a disorder process. Although there is considerable evidence that SIRT6 is a tumor suppressor, the effect is double-edged since it can also inactivate tumor suppression proteins FOXO3 and ABBA. Several SIRT6 inhibitors have been reported previously, blah, blah, blah. DNA damage, okay. Interestingly, catechins have also been demonstrated to protect cells against oxidative stress and DNA damage by increasing the activity of sirtuins. Okay, interestingly, the most prominent activators for SIRT6 among the flavonoids were the Anthocyanidins, the universal plant pigment responsible for the red, purple, and blue color. Remember, red, purple, and blue color in many fruits and vegetables and flowers. The most potent compound in the class of anthocyanins was cyanidin, significantly increased the deacetylation activity of SIRT6. It is the most abundant in red berries, including bilberry, raspberry, and cranberry. Studies have suggested that anthocyanidins, including cyanidin, may play important roles in helping to reduce the risk of many age-related diseases. The effect has been linked to their protective effect against oxidative stress, which results in the decreased production of free radicals and nitrogen species. Cell culture and in vitro studies of anthocyanidins and their glycosylated counterparts, anthocyanins, reveals anti-carcinogenic properties against colon, skin, and lung cancer, while laboratory studies have provided some insight into how anthocyanins may work. The exact mechanism for how these compounds prevent cancer is unclear. Thus far, studies in a variety of cancer cells revealed that anthocyanins activate detoxifying enzymes, prevent cancer cell proliferation, induce cancer cell apoptosis. So they stop the cancer from spreading, and they increase the rate that the cells die. Okay. To the best of our knowledge, this is the first study that showed the upregulation of SIRT6 in colon adenocarcinoma. Additionally, cyanidin affected the expression levels of SIRT6 associated with genes such as FOXO3 alpha, TWIST1, GLUT1. FOXO3 alpha belongs to the family of Forca. Okay, this is not important. I mean, it is important, but for our purposes, it's not really. We just need to know that these things seem to work. Sirtuins are involved in the number of central physiological processes, and their activities are likely regulated by endogenous signaling pathways in a tissue-specific and signal-dependent manner at various levels. Sirtuins can be regulated by many mechanisms, including transcriptionally and post-transcriptionally, by changing the stability, activity, localization, and degradation of the protein. In addition, protein complex formation and other binding partner proteins and changes in NAD+ availability may play an important role in regulation of sirtuin functions and expression. Many enzymes such as nicotinamide, are involved in NAD+ synthesis, which is upregulated by AMP activation, a kinase, AMPK. Polyphenols such as resveratrol and anthocyanins have been reported to activate AMPK, an effect that may be mediated by SIRT1. Thus, the activation of AMPK by polyphenols such as cyanidin may be one possible mechanism to upregulate sirtuin 6 expression in CACO2 cells. That was the colon cancer cells. Exercise and sirtuins, a way to mitochondrial health and skeletal muscle. I'm skipping to the conclusions, but you can obviously follow this. It'll be linked in the description. In conclusion, the effects of exercise on SIRT1 and SIRT3 in human skeletal muscle will depend on the type of exercise. The effect of acute exercise has been studied in both SIRT1 and SIRT3. The results indicate that SIRT1 and not SIRT3 is activated after a single load of exercise, apparently an effect of increased activation of AMPK. The increase in SIRT1 leads to an increase in PGC-1 alpha. The effect of exercise training on sirtuins has been studied with several sessions of high-intensity interval training or aerobic exercise. Investigations of high-intensity interval training have focused on analyzing the effect on SIRT1. These investigations provide evidence that the benefits of this type of training are regulated through the increase of SIRT1 and PGC-1 alpha. However, there is little evidence on the effect on both sirtuins. Meanwhile, the effect of interventions with aerobic training has been studied only on SIRT3. The results indicate that after several sessions of aerobic training, there is an increase of SIRT3 in sedentary individuals and increases of PGC-1A. In addition, NAMPT also increases as an effect of exercise, allowing the synthesis of NAD+, the main substrate of sirtuins. So this is how exercise literally de-ages you. It reverses the aging process. Due to the methodological complexity, the investigations have focused on some part of the metabolic pathway. The information we currently have is complemented by the results of all these investigations, even the animal and cellular models. It is known that exercise activates each one of the sirtuin pathways in different ways, but the outcome is common: to improve the mitochondrial health, which places them as therapeutic targets for the treatment of metabolic diseases such as obesity, insulin resistance, and type 2 diabetes. So in order to identify exercise training that can efficiently achieve greater improvements in these metabolic states, it will be necessary to perform more research that evaluates the effect of different types of exercise on both sirtuins and the health benefits in metabolically compromised individuals of different ages and different fitness levels. And that's going to be all for this video.